I would like to talk to you about HIV and this one, okay, and explaining you how nanomedicine might change the treatment paradigm of HIV. I am very fortunate that I had a very good introductory talk in front of me, so I don't have to introduce you the immune system or the importance of the nanomedicine. So, but I would like to talk to you this product. This is our nanomedicine, which is in the needleless container, and this is our application device. And all of these things has to work together to achieve safety and efficacy and targeting and novel therapeutic vaccines. So what is the challenge in HIV? We do not have a cure, and we do not have a vaccine, and this is a disease which is still a deadly disease. And the present treatment, we have almost 30 drugs to treat HIV. They are working very well, especially in combination. We use presently 3 or more drugs to lower the viral load. And we need to give to these drugs every day, otherwise resistance develops. But when you take it every day, you can live a long life. But recently there was a study which shows that even when you are fully controlled with antiretroviral drugs, you still live 11 years less than an uninfected individual. So it's still some challenge in HIV treatment. But we envision how nanomedicine could change HIV treatment is depicted here. What we are developing is a therapeutic vaccine which is administered by the patch. This would be a drug-sparing and immune-boosting therapeutic vaccine. We envision that we will give it 4 times a year at every regular visit when HIV-infected people go to the doctor anyway for the checkup. And this patch is on the patients only for 3 hours. After the patients at home can take it out, the treatment is done. Based on the results so far, this treatment has only transient local toxicities. I will show you, and provides an option for a cure, which would be a remission similar to cancer treatments. So, since I am talking about therapeutic vaccine, I would like to overview to you what is the difference between therapeutic and prophylactic vaccine. So when we are talking about general vaccine, you think about the vaccination of a healthy children, or perhaps traveler vaccine, which you are healthy, which you are given to protect you against a disease. Therapeutic vaccines are different. They are given to people who already have the disease, especially they have the deadly diseases, chronic diseases like virus infection, cancer, or allergies. So the patient population, the safety-benefit ratio is very different. Second, in a healthy person, vaccines are given to induce an immunity to an unknown pathogen. In therapeutic vaccine, you already have a lot of pathogen, you already have immune response, you have to boost or modify immunity. In prophylactic vaccine, the efficacy is based on antibodies. You have to protect the body from a pathogen to enter into the body. This is mediated by antibody. Generally, therapeutic vaccine antibody response is not effective anymore. You have to induce a T-cell response to kill cells which are causing the disease, like cancer cells or virus-infected cells. And finally, prophylactic vaccines has never worked as a therapeutics. So they worked in healthy people, they never worked after the person already get the infections. So we need really a new kind of technology for therapeutic vaccines. And we have already one cancer vaccine, which was approved last year by the FDA. This is a dendritic cell-based vaccine. Indeed, this is using a new technology, the new cell, the dendritic cells, to induce new immune responses in patients who already have cancer. And this is a technology, it's a very sophisticated technology. The way to target these dendritic cells, you have to take out blood from the patients, a significant amount of blood. You culture these cells for days, put the antigen onto the cells, and then characterize the cells, ship back to the doctor's office and treat the patient. So this is a huge logistic challenge to develop this, to treat the patients with this cancer vaccine. However, the efficacy is pretty good, actually better than the drug therapies. And the efficacy, the median survival benefit is 4.1 months. But what is also interesting that you can see here, I think this pointer does, yeah, you can see here that there is a delayed treatment effect. So early after vaccination, there is no difference between the placebo and the control and the treatment group. But after a long period of time, you can see this treatment effect here, which is this 4.5 months. This is very, very different kind of treatment than the drug treatment. So we are in the field very, very optimistic that this kind of new treatment will change the treatment paradigm of cancer. So why do we need nanomedicine for therapeutic vaccination? I have put here 3 important points why I believe we need nanomedicine. One is scientific reason, because we want to target the antigen into the dendritic cells. As Professor Hunziker told, nanomedicine is very important to target the drug to certain place, in this case dendritic cells, and decrease the amount of toxicities. The second reason is medical reason. Of course, we want to provide a safe, effective, and curative treatment. In this case, what I am talking about is HIV. And finally, business reason. The previous method, Dendreon, is able to treat in 1 year 2,000 patients. 2,000 patients is one vaccination treatment is 3 vaccine is $100,000. So in this cost, and we would not be able to treat 33 million HIV-infected patients. So what we are offering with the nanomedicine, what is what is the difference? So what we can do, and I will show you how, replace the 3-day ex vivo treatment with a 3-hour DermaVir topical patch, which patch contains our nanomedicine. And then the final mechanism is exactly the same. You target the antigens to dendritic cells, induce T-cell responses, and these T-cell responses, in the case of Dendreon, kills the prostate cancer cells. In the case of genetic immunity, DermaVir product, we kill HIV-infected cells. This is our nanomedicine. This is our pathogen-like nanomedicine formulation. This pathogen-like nanomedicine has 3 components. One is a plasmid DNA, and the plasmid DNA encodes the antigens. And the advantage of the plasmid DNA that we are not only able to put 1 single antigen or even 2. In this particular plasmid DNA, we encoded 15 antigen. You cannot do this with peptide or protein antigen. Then we put in our nanomedicine a polymer, a special polymer. We call it polyethylenimine mannose, and a solvent. And when we put a certain way together, we get a structure which looks like as a pathogen. This is about 100 nanometer big, and it has sugar residues on the surface. And then we put our nanomedicine into a plastic container. The advantage of this nanomedicine is not only that it looks like a pathogen, but also acts like a pathogen. So it targets the dendritic cells, The dendritic cells are thinking this is a pathogen. They like to pick up pathogen. And after they pick up, they enter in the endosome. This polymer, this polyethylenimine mannose, has the property to break the endosome and bring the DNA to the nucleus. It is very important for the expression of antigen and antigen presentation and induction of immune responses. So as I mentioned that we applied this pathogen-like nanomedicine topically. Why do we apply topically? Because we do not want to go to this ex vivo procedure. It would be too much cumbersome. And our idea was actually that the precursors of the dendritic cells, which we need to target, are located in the skin, actually in the surface of the skin, in the epidermis, called Langerhans cells. And we were thinking, how can we deliver this nanomedicine in vivo to the Langerhans cells? And we are just mimicking the natural way that what normally these Langerhans cells picking up the pathogens. So what happens is, first we have to do a skin preparation, which we do it. We have a method for that, and I will show you in the next slide. And that skin preparation does two things. One thing is just interrupt the stratum corneum, which allows the nanoparticle for penetrating into the epidermis. But perhaps even as importantly, in this epidermis, these Langerhans cells get these Langerhans cells, which is these cells which are look like as a neuron there, get activated. And after these Langerhans cells get activated, they are looking for pathogen outside. So what we do at this point, we put a patch. This is a semi-occlusive patch specially designed. And this is just a container containing our liquid nanomedicines. And indeed, then we put these nanomedicines under the patch. So then you can see these activated Langerhans cells ready to pick up the pathogen-like nanoparticles. And indeed, they pick it up. They get signals to mature, go to the lymph node, and present the DNA encoded antigen to CD4 and CD8 T cells. So indeed, we have shown, and I am not sharing with you this data, it is published, that this kind of dendritic cells, which we prime with the pathogen-like nanomedicine, can very effectively activate the immune system and induce both CD4 and CD8 mediated T cell responses. And after that, these cells, which are now antigen-specific T cells, are going around the body and looking for HIV-infected cells. And it doesn't matter that you have HIV-infected cells in the reservoirs or the brain or in the big toe, they find it and kill it. So what is the logistic of DermaVir immunization? What are we doing in the clinic? First, we do the skin preparation, as I explained to you. This takes some exfoliation and tape stripping, so to interrupt the stratum corneum. It is very important for penetration, as I told you, and also activating the Langerhans cells. The side effect of the skin preparation is a transient mild erythema, which we want to have because this is the activation signal for the Langerhans cells. Then we put the patch on the skin, and you see there is this opening. As I mentioned, this is just a container, and we put the liquid nanomedicine after the patch and close it. The patch stays on the skin about 3 hours, and then the patient goes home and removes the patch. Based on our clinical and immunological evidences, we think that every 3 months this kind of treatment has to be repeated. What about the clinical and safety data? We have done several preclinical studies and also 3 clinical studies. And basically, we were able to demonstrate that there is no difference between placebo and the treatment group. So this kind of DermaVir treatment is very safe and well tolerated. The only side effect which we repeatedly see is that this transient mild erythema. What about the clinical studies? I would like to review quickly 3 clinical studies which we have done. Phase 1 clinical study was done in Hungary on 9 subjects in 3 different doses, 1 single immunization, just to study the safety and preliminary immunogenicity. We always use HIV-positive patients. In this case, patients were treated with the state-of-art therapy. And what we were able to show, besides the safety, is that in every patient we could see induction of immune responses. These immune responses was kind of memory T-cell responses. And the best dose was actually the 0.4 milligram dose, the medium dose. And what we could also see that this immune response is optimum and at day 28 and after decreases. So this is not so surprising that these T cells have a certain half-life. So this also suggests that we need repeated immunization schedule. The second trial was with repeated immunization schedule in the same patient population done in the United States. It was a randomized, double-blinded, placebo-controlled trial. We have, again, found very good safety. And the major results of this trial was that we could see, again, antigen-specific T-cell boosted in this HIV-positive patients population. And again, the best dose was in the medium dose, the 0.4 milligram dose. It is very interesting. We always see this bell-shaped curve very consistently in every clinical trial that we have done so far. And finally, the phase 2 clinical trial was performed in Germany. We had 36 HIV-positive patients, but these patients were not treated with the state-of-art therapy. So they had HIV replication, free HIV replicating in the body. The viral load was between 5,000 and 150,000 copies per milliliter. So what we wanted to show here, not only the safety, but we wanted to have some preliminary data that we indeed can kill HIV-infected cells in the patient population, because in the heart-treated population, we cannot measure that. They are all undetectable. And indeed, we had a very encouraging data, because we were able to see about 70% viral load reduction in the optimal dose cohort, which is again the 0.4 milligram cohort. This is the cohort which in this population also had the maximum immune response. And also very interesting to show that this kinetic is very different than the drug kinetic. The drugs are acting very slow. This immune response it needs time to act. So this is very similar to the cancer vaccine, so it takes time, and we can see a delayed treatment effect. So how do we change the treatment paradigm? That it is after you having this clinical data. I would like to show you what we envision. There is not only technological challenge in HIV treatment and then most of the other diseases. There are other challenges, even when you already think that your technology is working. In HIV, there is a huge variability of the virus. As everybody knows that the virus, even in one patient, can change all the time, but it's changing based on territory and different regions. But it's not enough that we have a huge virus variability in HIV-infected patients. We have also genetic variability in the patient, because these epitopes are presenting on the HLA molecules, and the HLA molecules are genetically diversified in different individuals. So we have to deal somehow with personalized medicine. I have to somehow develop to this patient's personalized medicine. How do we envision that to do it cost effectively, as you asked from the previous speakers? So what we want to do, basically, we already know we have biomarkers. We can sequence the virus, and we can measure the HLA. This is already available. And what we want to do, we envision to have, actually we have already, a DermaVir product family, which we can manufacture commercially. This DermaVir product family contains the same HIV antigens, but in different, taking in consideration the HIV variability. For example, clade A, clade B, clade C, what is common in Africa, Asia, America. But you can measure the single patients, which kind of virus these patients infected, which kind of genetic HLA background it has, and we developed a software called eMiner, where you put the data, and the eMiner would be able to predict the epitopes which is presented in the patient's body by the patient's virus, and select the optimal DermaVir product from the DermaVir treatment family. And the doctor basically would prescribe this personalized DermaVir, which is available in the pharmacy, and treat the patients with that particular product. So that is still not changing the paradigm, even when it's effective. How do we want to stop the present drug treatment, and what is our evidence, or what is our vision? Why would we be able to stop that heart treatment? So this slide is explaining our present understanding of HIV infection. You have, in early infection, a lot of productively infected cells, showing in the left. It's not working anymore, but in the left side, in the upper part, you can see these productively infected cells, and the lower part, you can see the reservoirs. So in primary infection, the reservoirs are very slow. You just get infected. But very, very quickly, you have a lot of cells in the reservoirs. These reservoirs are consisting from cells which are not producing virus for whatever reason, and which produce a little amount of virus, like quiescent T cells or macrophages. And then you have also the productively infected cells. So what our heart therapy is doing, actually inhibiting the reinfection of the new cells. So basically decrease the viral load to undetectable, but by decreasing this reinfection, these productively infected cells. But the reservoir stays there. This is not influenced by the heart. Therefore, if you interrupt heart, you have the virus back in a very fast way. What we can do with DermaVir treatment, we not only eliminate the cells which are productively infected, but we can eliminate also the cells which are in the reservoirs and producing just small amount of virus. Therefore, we envision, and we already have animal data for that after stopping therapy, we might be able to control with our immune system the virus replication. How do we want to achieve that? Here is, we demonstrate what is going on in the patient. When you don't have treatment, you have high viral load, measured by HIV RNA. You have reservoirs, obviously. And then you have an immune response which tries to fight against the virus. Indeed, this immune response is not too bad, because if you are dealing with untreated HIV infection, the immune system controls is up to 7 to 10 years. When we treat with the heart therapy, presently state-of-art therapy, we decrease the viral load to undetectable level, as it shows by the blue line. We decrease a little bit of the reservoirs, but not too much. It stays there. And unfortunately, since we decrease the antigen amount, there is decreasing immune response. So what we want to do with DermaVir, that we want to add DermaVir on the top of first and the top of the heart. What we envisioned would happen, we boost the immune responses and decrease the reservoirs. So first we could spare some antiretroviral therapy, maybe one class of drug, which is the most toxic. And then after we achieved, when we achieved the reconstitution of the immune response and decreased significantly the HIV reservoirs, we envision that we could stop heart and the immune response could control virus replication similarly that in the elite controllers. So where do we see it in the market? yes, there will be a lot of generic drugs. And this is good for us, actually, because when DermaVir comes to the market, there will be more patients treated, and we could give DermaVir to more patients. And what would be the treatment benefit? That you could first stop the most toxic antiretroviral class. And what you could see fewer adverse events by decreasing the drug exposure and by maintaining undetectable RNA levels, and improving health outcome by limiting the development of drug resistance by decreasing the reservoirs. And I think that there will be some patients, hopefully many, who could stop all antiretroviral drug and would be fully controlled by DermaVir alone. Regarding the treatment paradigm, we envision that the DermaVir sale will increase because there is a larger treatment population, because, as I mentioned, there will be more people access treatment because the generic heart. There will be the trend, or there is already a trend for early treatment, and these people will therefore survive longer. So you will have a large patient population to treat, and there you will have a product which you could treat a large patient population. We see additional benefit over heart, as I mentioned to you, with sparing on antiretroviral therapy, improving the safety of the treatment, and there is a potential for remission. And we envision that this kind of immune therapy will be not too much expensive, so there will be insurance coverage for that as well. Finally, how do we do this? That was also a question, how is it possible? Which kind of business model you need to do this new kind of therapy very cost-effectively? This is our business model. We are a small company, and the same company we have discovery, preclinical, clinical trial, and CMC, and everybody is trying to work together to develop this new kind of therapeutic vaccine to provide a cure for HIV. And finally, this is my beautiful city, Budapest, where the company is located, and thank you for your attention.
Thank you very much, Juliana, for your very nice presentation. There might be some questions, I think.
Thank you very much for this presentation. A question I ask myself sometimes is, in a virus infection like HIV, you have active production of all the virus-associated proteins. What is the explanation that you see a kind of protective immune reaction against the synthetic vaccine of these proteins, while you don't see the same immune reaction due to the natural infection of the virus, where the same antigen is also circulating?
Yes, it is an excellent question, and I think it is the key question of therapeutic vaccine in HIV or cancer. When you have a lot of antigen, why would a therapeutic vaccine work? We are not giving additional antigen, and I think vaccine which just inject additional antigen cannot work because you have plenty of antigen already. But you have problems with antigen presentation. In chronic virus infection or cancer, somatic cells present this antigen, and these somatic cells can only exhaust immune response which is already generated because they already make memory cell proliferation. But what dendritic cell can do, they increase the memory pool. Then this is only the dendritic cell can do. Therefore, the new technology, which we target specifically the antigens to dendritic cells by ex vivo with the topical application, is essential for therapeutic vaccine. So if you can increase the memory pool by putting the antigen into the dendritic cells, not only somewhere in the periphery, targeting the antigen to dendritic cells, I think this is the key.